So we're looking at the basic unit of life. All living things are composed of cells. We know this. And so you've probably studied the cell in life science in the past. And so this unit is aimed to review and learn more, dig deeper into the cell. So let's review the facts about living things. What are the main characteristics of organisms? So you can hear a little yippy dog in the background, and that's my neighbor's dog. Anyhow, we know that all living things are made out of cells. We also know that in order to identify a living thing requires energy or food. They reproduce. They maintain what we call homeostasis, or an equilibrium, or balance with the external environment. We know that they are organized. We know that they respond to the environment. We also know that they grow and or develop. And one of the last things is that they exchange materials with their surroundings, whether it's water, wastes, or even gases. So as we look at the organization of things, we're going to start with the non-living levels that we've already studied here in biology. And we've studied the atom, which is the basic unit of any element. It is the smallest particle that still retains the characteristics of that element. A molecule, excuse me, are compounds like carbohydrates and proteins that involve multiple elements. Then the next level up would be organelles. Now, organelles are considered non-living because by themselves they can't demonstrate all of those different characteristics of life. So things like the nucleus, the endoplasmic reticulum, and the Golgi complex. When we look at the living levels, the cell is like the atom. It is the basic unit of all living things. It makes up all organisms. Tissues are groups of cells that work together, and then organs are groups of tissues that work together. When we put organs together, we get organ systems. And then once we have organ systems put together, that makes up a full organism. And I should explain that at that point, it's considered a multicellular organism. All right. When we take the organisms, thinking back to our ecosystems unit, we take those organisms and they make up populations. Populations, all of them in an area, make up a community. An ecosystem then is that physical environment, the habitat, as well as everything living in it. Then there's the biome, and that's going to include the physical factors like precipitation and temperature. And then there's the biosphere, where all living and the non-living things exist on Earth. So, let's look into the history of cells in what we call the cell theory. In 1665, Robert Hooke used a microscope to examine a thin slice of cork. Cork, you may or may not know, is dead plant cell walls. So what we're seeing is the external structure, skeleton, if you will, of those plant cells. So it was that long ago that those were first observed. What he saw looked like small little boxes, okay? Now, he was the first person to actually see these cells. Hooke was responsible for naming cells. He's the one that gave them their name. He called them that because they reminded him of the small rooms that he, as a monk, lived in. So he was from Austria. He was a monk at a monastery and had the ability and capability to study such things. Now, Anton van Leeuwenhoek, in 1673, who was the Dutch microscope maker, and he was the first person to develop the first microscope. That's how we were able to even observe cells. was the first person to view what we consider a living organism. And the way he did that was he used his little handheld microscope, which looked just like this. It was basically a brass screw, okay? And then up here on the end, there's a little tiny needle point, and that was what he considered his sample holder. That was the stage, if you will. And then there was the little tiny ground lens right there. So he would put whatever it was he wanted to look at right there on the end, then pick this up, turn it around, and look from the other side through the lens in order to see his sample. And he started with pond water, believe it or not. And so this little guy over here is one of the organisms that Van Leeuwenhoek first saw. Now, he also even looked at his scrapings of his teeth. Gross. Didn't brush his teeth very much, apparently. So, how did the theory of the cell actually begin? Well, in 1838, a German botanist named Matthias Schleiden concluded that all plants were made out of cells. He used that simplified microscope, or a simple microscope, and was able to observe that all plants were composed out of the same thing. So he's considered a co-founder of the cell theory. In 1939, just 1 year later, a German zoologist named Theodor Schwann concluded that all animals were made out of cells as well. Exact same discovery, except one was for plants and one was for animals. So Schwann is also considered a co-founder, not co-founded, a co-founder of the cell theory. So this was the beginning of the cell theory. Now in 1855, based upon those two discoveries, a medical doctor named Rudolf Virchow observed under the microscope, cells actually dividing. And over here we can see these are the nuclei of those individual cells. And he reasoned that all cells come from other pre-existing cells by cell division. So this is the cell theory. First, we know that all living things are made of cells. That's the first postulate. We know that cells are the basic unit of structure and function in an organism. They are the basic unit of life. And that all cells come from the reproduction of existing cells through what we call cell division. So since that time, there have been some discoveries regarding the cell. And one of the major theories that has come from the cell theory was by Lynn Margulies, and she developed what's called the endosymbiotic theory. So this was in 1970, fairly recent, not obviously when you guys were born, but she provided evidence that some organelles within cells were at one time possibly free living cells themselves. So their supporting evidence included in their organelles. Their organelles actually have their own DNA. So they include the chloroplasts and the mitochondria found in both plant and animal cells. So the idea was that over time, these ancestral cells right here, we would consider them bacteria or prokaryotic cells. As they changed and evolved over time, there was a folding in of their cell membrane. And as that cell membrane folded in, it wrapped around the nucleus. Now, within that cell, there's the mitochondria, right? And the chloroplasts. And so if this was a photosynthetic bacterium, it had both, okay? And in this cell, if it was a heterotroph, it only had the mitochondria. Now, if while it was ingesting other things, it accidentally released some of the mitochondria, or took in the mitochondria, those mitochondria then were in that cell, and so as that cell reproduced, it passed down its mitochondria, meaning that all the future offspring had that particular organelle. So kind of the same idea existed in those photosynthetic. So if this was photosynthetic, that chloroplast, and one of these bacteria took it in, then that bacteria continued to pass on that plastid for generations. So kind of interesting. So we're going to do a little bit of research about the endosymbiotic theory and see if we can't find some articles related to it. So that's the history of the cell theory, and we'll be moving on soon.